Hello students. So in this video let us discuss about the experimental proof, very important experimental proof for the semi-conservative DNA replication. So in previous video we discussed about DNA replication which is semi-conservative. So theoretically we explained that, so in a daughter DNA molecules there is a one strand comes from parent and another strand is synthesized newly. So which is comes from parent, that is called template, and which is synthesized newly we call it as complementary, right. So but experiment how it is said to be parental and how it is said to be newly synthesized, how can we identify it? So experiment is required here. So for that the two scientists, so that is Matthew Michelson and Franklin Stahl. Matthew Michelson and Franklin Stahl. So in 1958, so they gave experimental proof for the semi-conservative method of DNA replication, right. How it is semi-conservative, and they are explained. So by using some organisms, especially it was first time experimentally proved in E. coli bacteria, right. So that was explained in Escherichia coli, E. coli, one of the familiar bacteria it is, which is found in the colon of human beings also. So that is what we call it as E. coli, Escherichia coli, right. So this organism is used by these scientists and they are proved it is a semi-conservative. The organism used is E. coli bacteria, but first time it is started in a bacteria, later on even in higher organisms also it is proved. So that is in plants, and that is a faba bean. So it was explained again by Tyler, okay. So and even in human beings, human cells also it is explained, right, it is experimentally proved, right. So now the medium used for the synthesis of this DNA, that is the medium with NH4Cl, nitrogenous compound it is. So that is one of the ammonium chloride which is containing nitrogen. Nitrogen is one of the important part of DNA molecule, as you know, because nitrogen bases are formed by nitrogen, right. So this nitrogen is used here, here the isotopes of nitrogen is normally which has 14 number, right, 14. So this is the N, what you call N14 is a normal one, and if it is said to be N15 here, so that is isotope, heavy isotope. So this is used as a heavy isotope, right. This is the heavy isotope. So from this the NH4Cl, that is ammonium chloride, so which is there in that media, which they used the media for the culture of that bacteria, right. So and another chemical also used here, cesium chloride, what you call cesium chloride. So that is the CSCl, cesium chloride, because this is depends on this experiment is depends on the heavy isotope of nitrogen, because the after centrifugation heavy molecules are settled at the bottom in the cesium chloride is used here, right, because it may not react with this one. So that is why it is used here, right. So cesium chloride is only for centrifugation or separated, so that DNA is separated on that cesium chloride, right. So next here the steps are involved in this experiment, that is E. coli, first step, in the first step E. coli grown, they are grew the E. coli on the medium which containing this heavy isotope of nitrogen, that is the we are not using here like free form of nitrogen. So, that is should be in a compound form. So, that is the organisms utilizing form, that is said to be organisms how it is utilized. For example, we need a iron, we are not going to eat the iron. So, we are eating the iron means in the form of food, right. So, in the same way the bacteria will utilize this as a raw material for its synthesis of DNA, right. So, that E. coli bacteria are cultured, they are starts culturing this E. coli, that is in the this medium containing yes NH4Cl, that is which with the heavy isotope, that is what heavy isotope. Nitrogen 15 is the called heavy isotope, nitrogen 14 is a normal isotope which is less in weight, which is heavy in weight, right. So, density gradient we can say, density gradient, based on the density gradient. So, it is centrifuged, later on we will get the result, right. So, next, so first they are starts growing the these E. coli in this medium, right. Finally, they will get yes bacterial cells with yes the DNA which is containing heavy isotope, right, heavy isotope. After that they starts after several generation they do the centrifugation mechanism. They used this cesium chloride for centrifugation and they observed it in the centrifugation. So, that is in the just a minute. So, I take here first what they will do, the centrifugation. So, I will take before that centrifugation the diagram of DNA. The both strands are containing this both strands which are containing N15, right. So, this is what the N15 because in this strands nitrogen bases are there, right. So, this strand DNA contain, this DNA molecule contain heavy nitrogen, that is a heavy nitrogen, that is isotope, right. It is not radioactive isotope, remember it is not radioactive isotope. So, another experiment may be done by other scientists, so with radioactive isotope, right. But here this is a not radioactive isotope, it is a heavy isotope which is in a nitrogen, right. So, now after several generation, so these strands containing heavy isotope as a nitrogen, right. So, this is centrifuged, what they did centrifuged, these cells are when centrifuged, right. So, this is the, you can consider it as a test tube here, just a minute. Yes, okay. So, here it settled at bottom, right. So, this is settled at a bottom, here they observed it in the cesium chloride is somewhat transparent. So, this may be a cesium up to here, this is the cesium chloride you can consider. So, this is CSCl and this is what yes when they centrifuged. So, that it appears at the lower side of the test tube, right. So, that remains here, that is what you call after several generation. So, usually as you know the bacteria takes about for multiplication 20 minutes, right. 20 minutes is enough for the multiplication. After 20 minutes the second generation is formed, right. After 40 minutes the third generation, right. Every 20 minutes they can get multiplied, the reproduction is possible, the next generation is formed, right. So, in this way, so these bacteria are used here and they are even human friendly, right. They are not disease causing, they are not causing any diseases. So, that is why E. coli is commonly used for the many experiments. So, even the Matthew Michelson and Franklin Stahl also used the same bacteria, that is E. coli here. So, this E. coli is now centrifuged. After that, due to presence in the E. coli which DNA is developed. So, that is the DNA containing heavy isotope, hence when they centrifuge this one, centrifugation. So, simple device is there, simple machine. So, in that when they centrifuged it, so in the presence of cesium chloride, so there is a test tube, right. This is what the centrifugal tubes also we can say. So, this is what centrifuge tube. So, where this region, so not at middle, at the lower side it is occurred, right. So, next what they did. So, why it's become so? See, after several generation, what happens in the second step what I have taken here, the heavy nitrogen isotope was incorporated into newly synthesized strands, that is incorporated into the newly synthesized strands, right, of DNA. The whatever DNA, so actually it was a normal DNA. So, it was normal DNA, yes, which was a normal DNA in that bacteria. So, next it's become maybe partially, so right. So, then again after several generation, so again it's become a, yes, so like this, right. So, finally, so all the molecules, all the DNA molecules present in the after many generations, so totally that's become as N15, that is a heavy isotope, right. So, now this is the what we call first it is centrifuged and they will get result here at the bottom, right. So, next the why it has become bottom, because the nitrogen is heavy and that is incorporated in newly synthesized DNA, means in the new cells of bacteria, right. So, next, so transfer of these cells they are not killing here, these cells are transferred. So, right, these transfer of cells in the normal medium means not having any heavy isotope here, normal isotope, that is N14. So, that is allowed to multiply here. So, after 20 minutes they are, of course, they are get multiplied their numbers, okay. The multiplication of cells is occurs after 20 minutes, right. So, this is what we can consider the first generation here, right. So, this is what after 20 minutes we consider it as a first generation, right. So, the several generations are done before the starting this experiment, right. So, they are developed in heavy isotope medium, but here in normal isotope after first generation we can say, after allowed to enter in the allowed to grow or culture, that is in the normal medium, the medium which is containing normal nitrogen, that is called normal medium, right. So, that is represented by N15 NH4Cl, the same ammonium chloride. So, that is with the normal nitrogen. So, right, that is not heavy, that is said to be light compared to the heavy, right. So, this is the normal one, light one, right. After 20 minutes what happens, the samples were separated, right. After 20 minutes the samples were separated. So, here what happens, this is sample were separated on the cesium chloride here, separated on the cesium chloride with the help of centrifugation. Again here they did the centrifugation. So, in this centrifugation what we observed here, so that experiment, so what they observed is, see, that is observed as the medium, see they observed here in the middle and even something light color also, right, they observed here. So, this is what the what we referred in the result they will get here after first generation. This is what the after 20 minutes means, that is the first generation. The DNA extracted had hybrid, that is here N15 and N14. So, here N15, here that is N14 we can consider, right. So, this is what the due to 50% is heavy, 50% is normal. So, due to that, so it is not go to the bottom, not here. So, it comes at the middle, right. So, based on the density, so it will comes at the middle of that test tube. So, this is what as well cesium chloride. So, we can show it as cesium chloride. So, this is what the here it appears. That is what we referred it as a hybrid or referred as intermediate density. So, they are appeared in intermediate density, that shows the intermediate density. This is the heavy density here at bottom, at middle that is showing intermediate density, right, intermediate. So, next, due to presence of 50% heavy and 50% normal, right. So, in the after second generation means after, so which is allowed to after 40 minutes, right, 40 minutes. Next, after 40 minutes the same bacterial cells are allowed to centrifuge, right. So, when they centrifuge, what happens here, this is as usual the test tube we can show, right, cesium chloride. Next, here what they observed, so next here also 50% they observed, and here also they observed that, yes, intermediate one. So, that is here they observed N15 and N14, here only N14 and N14. N14 means here in the above, see, density is less of that 50% of cells are appears in this area and 50% are appears in this, right, in the middle of that cesium chloride, right. So, this indicates that, so there is this was what we called completely having both strands having, yes, heavy isotope, and then it allowed to grow in normal media. So, what happens, one is become, they get two separate strands, right. So, that is with two separate strands, and there is a formation of new strands, right. So, in this way strands are newly formed, right. So, new strands are formed here. See, again in the normal conditions, if they are allowed to another after 20 minutes, it is 20 minutes. Next, after 40 minutes, what happens here? There are 4 strands are formed. So, I will take little more space here, right. So, what happens here? These are again this forms a 2 daughter DNA after 20 minutes, and after 40 minutes what happens? After 40 minutes these strands become 4, right. So, ultimately what happens here? So, I will show these separate strands here. This is referred as normal here, right, and here it is, right. So, one more strand is here, right, and this is the normal strand. So, these are from parent strand with the second generation parental strands, So, that is, this is 14 N14. So, this is also N14, right. So, this is N15, this is also N15, right. So, what happens here? New strands are formed with normal, so which is developed in normal medium, the medium containing normal nitrogen, that is not heavy isotope. So, what happens here? New strands are formed, right, because all they are incorporated with the normal nitrogen, right. So, then what happens? So, these newly formed strands, what happens? Newly strands, the 50% of new strands appears in here, yes, 14. This is the both are said to be 14 here, okay. 14, this is also 14, N14, here also N14, both strands of these daughter cells, right. So, daughter molecules, right. So, and here 50% is 50%. So, there is no heavy, completely heavy as we taken here first one, right. So, first what we started with nitrogen 15, that is heavy isotope, Okay. So, in this way, by this experiment what we conclude, so again 50% we observed, yes, 50% we observed that heavy isotope in the daughter DNA molecule in the second generation. What is that? So, in the after second generation DNA extract, the DNA extract, this is what the second generation DNA extract, where so we observed that 50% means equal amount of the heavy DNA molecules and light DNA molecule, heavy or intermediate or what we call. So, that is what we call, yes, hybrid or DNA. Hybrid means here one is heavy isotope, another one is normal. So, that is here we observed. This is what the two strands are. So, with the heavy hybrid, and these are as usual normal strands, right. So, this is what we referred here, it as one of the experimental proof for the semi conservative DNA replication, right. So, I hope you will understand this. So, try to observe the steps here. So, first they starts developing the or growing the E. coli in a medium containing, yes, previously it was not, it is a normal, okay, that bacteria contain DNA, no, that DNA having the N14 in their DNA, nitrogen is 14, that is not isotope, that is normal DNA, right, normal nitrogen, right. So, after that, that is when grown in the isotope containing what we call medium. So, that is completely converted into the DNA is become a heavy, right. So, heavy molecule. So, based on this density, so they conducted this experiment not by radioactive isotope. So, actually radioactive isotope was conducted by radioactive thymidine by Tyler and his colleagues. Radioactive thymidine, as you know, thymidine, okay. So, instead of nitrogen they are used the thymidine. So, this is the become radioactive by Tyler and his colleagues, right. They also conducted the experiment in Vicia faba, that is a faba bean. So, we call it as faba bean. So, I hope you understood this experiment. So, please follow the steps, so and write the notes. Thank you, students.